Thank you.
I'm going to reclaim them now.
And you can
Got it. Yep, it showed up. Thank you.
Yep. All right. Okay. I think we're ready to go. Thank you very much for coming to the available room on campus at 1 p.m. on a Tuesday. You made the journey. All right. So you are all in for a treat with Allie Osborn's defense. I'm not going to go through the story of this defense. Allie is going to hit that. But I think of your time here as a lesson for patience, in which the stressed out Allie we all met in the beginning, look how calm she is. So yeah, and I think that also her committee could also agree that we've all been stressed out, not by Allie, but by biology, which does not ever do what you think it should do, which is why you should all do computation. But nevertheless, yeah, so Allie is the end of the grade 8 that defended from my lab, and definitely had unique challenges and excitement, and was willing to kind of take a chance. And everything you're going to see today, she had no idea how to do 2 years ago.
Yeah,
a little more than 2 years ago. So it is. I won't talk anymore, but I'm very thankful you all came, and I look forward to the presentation. So Allie Osborn.
well, thank you all for coming to my presentation. I very much appreciate it. Today I'll be talking about the evaluation of bacteriophage infection of Enterococcus faecalis in a monolayer. First, I want to introduce you to the Enterococci family. So Enterococci are gram-positive bacteria that are cocci in shape, meaning that they are spherical. They also occur in pairs or short chains, and they live in the environmental niches of the gastrointestinal tract, hospitals, streams, seawater, and soil. The most common enterococcal species within humans are Enterococcus faecalis and Enterococcus faecium. I specifically will be looking at Enterococcus faecalis today. When it is normally found in the gastrointestinal tract, but it can move to other parts of the body, and that is when it becomes an opportunistic pathogen. One area in which it can spread is the bladder, and it can cause UTIs. In fact, E. faecalis is associated with nosocomial UTIs, which are hospital-associated UTIs, and 20.1% to 61.5% of complicated UTIs and 15 to 30% of catheter-associated UTIs are caused by E. faecalis. Also, E. faecalis is the dominant species in recurrent UTIs, which are when a patient has 3 or more UTIs within 12 months or 2 or more UTIs within 6 months. So with this recurrent UTIs, there's often this antibiotic resistance, and that is seen in E. faecalis. So E. faecalis has this antibiotic resistance. It is intrinsically resistant to cephalosporins, aminoglycosides, lincosamides, and streptogramins. It also has resistance to other antibiotics. It just is geographically dependent, meaning what antibiotics are used in different locations determines the antibiotic resistance. These antibiotic resistant genes are carried by plasmids, which is circular DNA that is separate from the bacterial DNA and can also replicate independently. So with that, the emergence of antibiotic resistance, there is need for new treatments, one of which is phage therapy. So phage therapy involves the therapeutic administration of phages to treat bacterial infections. I will explain what phage are shortly. They are used, Frederick Twort discovered them in 1915, and Felix D. Horelli also discovered them in 1917, so independently of each other. They are used for treatment against bacteria until Fleming discovered antibiotics in 1928. However, recently there is this current rise in antibiotic resistance, so there is a renewed interest in phage therapy. So phage are viruses that infect bacteria, and they can be separated into 2 morphological groups, tailed and non-tailed. I specifically will be looking at tailed phage, which can be in 3 different types. So we have the syphilis, the myovirus, and podovirus. Each of these phage contains a capsid, a tail tube, a base plate, and then tail fibers, with the syphilis having a long and flexible tube, Potavirus also having a flexible tube that is much, much smaller. And then the myovirus, which has a tail that is covered by a sheath, making it less flexible. So phage also go through two different life cycles, the lytic and lysogenic cycles. So phage will randomly attach to and insert their DNA into bacterial cells. That bacteria is then the phage is inside the phage DNA is inside of the bacteria. And then the bacteria starts synthesizing and assembling new phage, producing phage DNA and proteins. Once enough phage have been created, a bacterial cell will burst or lyse, releasing fully formed phage that can then infect other bacteria surrounding that original bacteria. In the lysogenic cycle, we have the phage still inserting its DNA into the bacterial cell. That phage DNA then integrates into the bacterial chromosome, creating prophage. That bacteria is then able to replicate as it usually does with each new replicate containing that phage DNA within the bacterial chromosome. We specifically will be working with the lytic cycle because the phage we are working with are obligately lytic, meaning they only go through the lytic cycle. So now that we know what the phage are and what we're working with, I wanted to kind of give you an overview of how phage therapy works. So we have a patient that is infected with antibiotic-resistant bacteria. That bacteria is then isolated and many phage are tested against it to see if there are any that are able to lyse the bacteria. Once they have been phage have been determined to do so, those phage are purified and they go through genomic screening to look for genes that make it a positive use for phage therapy. One gene that isn't useful is lysogenic genes, because that makes them more likely to go through the lysogenic cycle, so they are unable to infect and kill the bacteria they are interacting with. Once done with genomic screening, they go through animal model and in vivo testing, and then they can be injected into a human patient for treatment. Some advantages of phage therapy are that they are host specific. So the phage are able to infect certain species, and within those species, it can only infect so many strains, so it cannot infect all of the strains that are available. They are abundant in nature, and then they have this ability of auto dosing. So when the phage are injected into a patient, they can infect and lyse bacteria, releasing more and more phage without there being a need for more insertion of phage, more dosing. So that leads us into our next question. Are there phage capable of killing many E. faecalis strains? And we first had to have our bacteria and phage that we were looking at. So we have a total of 22 bacterial isolates, with 16 of them being.
Hit the black little button, remote.
This one?
No, the one in your hand. There's a blackout button.
No.
I lied. Remote is working. What happened?
Oh. Intermission.
That's weird. It's like the OneDrive.
Yeah. So not connecting to the internet anymore? What are we doing?
No, you're connected to the internet. Where's the refresh button?
It's not even giving me, it's just an X.
Go back to OneDrive, the other tab. And at the display. There we go. Okay, now you got to share it.
Yep.
There you
Okay.
go. Okay.
So as I was saying, we have a certain amount of phage and bacteria that we are working with. There are a total of 22. I'm gonna do the thing again. Okay, now I hope there's no more issues with it. So we have a total of 22 bacterial strains that we are working with, 16 of which are bladder isolates and 2 of which are urogenital isolates. We also have one that is a blood isolate, with 3 others being knockout strains of that blood isolate, meaning that a gene has been removed that makes it different from that original blood isolate. We also have 3 phage isolates, 2 are urogenital and 1 is sewage. So that we know all of our bacteria and phage strains and isolates, we had to figure out the host range. So the host range is the range of bacterial strains that phage can infect and lyse out of a specific set of bacterial strains. So we figure this out through plating a lawn of bacteria onto a plate, and then we add drops of phage, allow that to grow overnight. And if there are these plaques, which are zones of clearing, we know that the phage have been able to infect and lyse the bacterial strain. If there are none of these plaques, then there is no lysing that is occurring. So we did this with our phage and our bacteria, isolates and strains. And as a reminder over here, just so you know, the yellow is the cells that lyse the bacteria. They are the host, and then not lyse are the ones that did not kill the bacteria strains. They are the non-host. So as you can see in this image over here, we have more non-hosts than hosts, but we did see that VD13 was able to have 9 hosts, had the most. VD1884 had 2, and myovirus 1 had 5. And interestingly, there are no overlapping hosts. So that means they each had their own unique host range. So once we figured out our host range, we decided to go with our just four of these bacterial strains for the remainder of our research. We chose these 4 because they were able to be lysed by at least one of our phage. Which then leads us into our next question: can phage attach E. faecalis strains so that they don't lyse? In order to visualize this, we are using transmission electron microscopy, or TEM. This allows for high resolution imaging of bacteria and phage independently and together. We first wanted to image everything independently. And so to do so, we took either our E. faecalis or phage, whatever we wanted to look at. We added a drop to a grid. We let that sit on the grid for a minute before drying it off with filter paper. We then added a drop of water to remove any unattached phage or bacteria that we were working with. Let that sit for a minute. And then we dried again with the filter paper, added a drop of uranyl acetate to stain whatever we were working with, bacteria or phage. Let that sit for a minute, dried it off, and then we let it sit for a full hour to dry before we imaged on the TEM. So our first image here is of E. faecalis strain UMB 1527. We did not take an image of all of our bacteria strains because they all should look the same. So we wanted to just visualize at least one of them. So here we were able to see our bacterial strain. We also did the same for all of our phage. So this is phage isolate myovirus 1. And it's just to reorganize your thoughts on how the phage look. We have the capsid up here. We have a sheath over the tail, and then we would have the base plate here and then a tail spike. And because there is that sheath over the tail, this is a myovirus phage. For the next one, VD13, we had this elongated capsid, but we did not have the sheath over the tail. And since it is a long tail and flexible, we know this is a syphovirus. The same can be said for VD1884, with there being no sheath. And so we can see the tail, it is flexible. It is also a syphovirus. So then we wanted to image the bacteria and the phage together. So to do so, we took our phage lysate and we combined it with a 1:50 ratio of E. faecalis to media. We left it in a tube for about 5 minutes. Then we added a drop of that mixture to a grid and let it sit for a full 20 minutes, just to allow for everything to attach to the film that's on that grid. Then we went through the same process that we did before with the removing of unattached bacteria and phage with the water, and then staining with the uranyl acetate before imaging on the TEM. So what we expect to see is, for the host, we'll see the phage attaching, and we'll see it attaching to the bacterial cell by its tail. For the non-host, we expect to either see the phage nearby around the bacteria or not present at all, but we will not be seeing any attachment. And this is what we expected going into this imaging process. So our first 3 images will be of the host. So we have VD13 with its host UMB 9329. Our black arrows here will represent unattached phage, and our white arrows will represent attached phage. So as you can see in this image here, we have an unattached phage up here, and then several attached phage all along the bacterial cell here, which confirms that there is attachment for BD13 and its host. Same was seen with BD1884 and its host. We saw these two here, and as I'm seeing now, there might be a little guy over here, but we are able to see attachment, so BD1884 also can attach to its host. However, for myovirus 1, with our red arrow here, we saw it present near the bacterial cell, but we never were able to see it attach. And we actually ran this test multiple times, but we never were able to confirm attachment. So myovirus 1 could not attach to its host. Then we did all of our non-hosts. So we took BD13 and combined it with its non-host UMB1527. And actually all around the bacterial cell here, we can see attachment of these phage. So there actually was attachment by the phage to the non-host. We also saw the same with BD1884. We saw attachment all along the bacterial cell, as well as an unattached phage over here. So again, BD1884 can attach to the non-host. And then of course, myovirus 1, we didn't see any phage anywhere near the bacterial cells. So there was no attachment from myovirus 1 to the non-host. So then overall, we know that there is phage attachment to host and non-host for BD13 and BD1884, and that myovirus 1 had no attachment for host or for non-host. But because of these images, we can confirm that host range does not represent if there is attachment to the host or non-host or not. Because we didn't see plaquing with the non-host, but we did see attachment. So that leads us into our next question. Can phage insert their DNA into E. faecalis strains that they don't lyse? And to do this, we're using a confocal microscope. So a confocal microscope uses light to create high-resolution images of cells and tissues. It can capture multiple 2D images at different depths that can be mapped together to form 3D images. This microscope uses lasers to fluoresce any organisms that have stains on them. So that is how we're going to visualize our bacteria and phage. So our first process was just to image the bacteria. So we took our E. faecalis culture and we grew it overnight in the incubator. We then took some of that culture and added to a poly-L lysine coated cover slip so that it can attach to the cover slip. We let that sit on the cover slip for 15 minutes at room temperature before we dried it off, and then added 4% PFA or paraformaldehyde, which fixes this bacterial cells in place. That was left on the cover slip for 10 minutes before it was dried off and then washed 3 times with sterile PBS. And then we added a DAPI mounting media, which DAPI is a blue stain that stains the nuclei, what it's interacting with. So in this case, the bacterial nuclei. Then we mounted the cover slip to the slide, sealed it with nail polish, and imaged on the confocal. So just to give you kind of an idea of how these images are going to look, I will have a laser up here above the image that will show you which lasers are on when I'm looking at the picture. I have the name of the bacteria or whatever I'm looking at up in the top right, and then the stains, the color will match with the image, and you will be able to know what they represent. So in this case, the blue is the nuclei. So from this image here, we were able to actually visualize several bacterial nuclei, meaning we could visualize the bacterial cells. Then we wanted to image the bacteria and phage together. So we first had to prep our phage. So we took our phage lysate and we combined it with cyto13. Cyto13 is a green stain that stains the phage DNA. We took that mixture and mixed it end over end for 15 minutes. We then put it through a filter and measured the fluorescence intensity on a plate reader. That fluorescence intensity had to be over 1,000 RFUs, or relative fluorescent units, for us to confirm that there will be fluorescence in our image. Then we prepped our bacteria as pretty much the same way as we did before. Then we took our phage lysate and cyto13 mixture and our E. faecalis, and we added it to the cover slip. We let that interact for 30 minutes before we went through the same process as before with the fixation, the washing, and then the DAPI mounting media added. So the first question we wanted to figure out was, how long does it take for phage to inject DNA into E. faecalis? So as you can see in the image here, we have the blue and green lasers on and the green laser on here. This is the same image here with different lasers on, with the nuclei and the phage DNA marked. So as you can see in this image, we had clear phage DNA insertion, and we actually tested this after those 30 minutes of interaction. We did this as well for an hour, but the slides did not turn out. So even with that issue, regardless, we are still able to see phage DNA insertion at 30 minutes, so it's not needed to be replicated. Then we wanted to see the entire bacterial cells along with that phage DNA and the nuclei. So we added this wheat germ agglutinin to stain the membrane. So WGA is a red stain that stains the bacterial membrane that or whatever it interacts with. So we took our WGA, we centrifuged it to separate anything that was stuck together. We added the WGA to SPBS, and then for that mixture, we added it to the cover slip and let it sit for an hour at room temperature, covered to avoid any bleaching of the stain. We then washed it 4 times with sterile PBS, and then went through the same mounting process as before, with us knowing that this process occurs right after fixation. So now that we had figured out our colors and how to stain everything, we are able to see the WGA. So the next 4 images are going to be exactly the same with different lasers on. So in this image, we just have the red laser on. And as you can see, we can see the membrane of all 4 of these bacterial strains here, or bacterial cells here. So we know that WGA is working. So then we turned on the green and red laser. So we can still see the membrane stain here with that green fluorescence in the middle showing that phage DNA insertion, with the yellow being where the red and the green stains overlapped. Then we had the green and the blue laser on. So what we can see here is we have the bacterial nuclei in all 4 of the bacteria, and then again, that phage DNA within the bacteria. Then we wanted to turn all 3 of our lasers on. And so we can see the membrane of the bacteria, the nuclei, and then the phage DNA within the bacteria. So we were able to see that all 3 of our stains work, and we can see that phage DNA insertion as well. So then we wanted to do host and non-host imaging, and we did the same thing as we did with the TEM with the same pairings as we did. So what we expect to see for the host is that the phage will insert their DNA into the host, and this will be how the bacteria look like. We'll have the nuclei of the bacteria stained in blue, and then we'll see that phage DNA insertion in green inside of the bacterial cell without that membrane being stained, because we were not having the WGA being added. For the non-host, we'll just see that bacterial nuclei without any membrane, without being able to see the membrane, and then there will be none of that phage DNA insertion occurring. So again, these are the first 3 are hosts, and then the next 3 are non-hosts. So for VD13, we were able to see clear phage DNA insertion. So we can confirm that there is VD13 can insert its DNA into its host. Same can be said for VD1884 and its host. So we again see that phage DNA insertion, and it matches up with where those nuclei are sitting. So that helps even further confirm that insertion. And then for biovirus 1, we actually also saw phage DNA insertion. So we were able to see phage DNA insertion for all of the phage and their hosts. For the non-hosts, we also saw this phage DNA insertion for VD13. So VD13 can insert its DNA into the non-host. Same could be said for VD1884. We were able to see that phage DNA insertion, a little bit more messy than the one previous, but we know that it matches up with the nuclei. So there is phage DNA insertion for VD1884 and the non-host. For myovirus 1, we do see some of this green fluorescence over here, but it's not to the same extent as the other images. So we can confirm that myovirus 1 cannot insert its DNA into the non-host. So then overall, we know that VD13 and VD1884 had DNA insertion into host and non-host E. faecalis, but myovirus 1 had insertion only into the host. So now comparing the TEM and the confocal images, we know that the TEM cannot capture the entire infection process because myovirus 1 was not able to, we did not visualize attachment for the host, but we did see phage DNA insertion. We also saw phage DNA insertion for VD13 and VD1884 non-host, and there was no phage DNA insertion for myovirus 1 non-host. So then we got to our next question, which is, can E. faecalis strains of interest attach to U2S cells? So U2S cells are human osteosarcoma cells, which means they are tumor cells. these cells grow quickly and they have an epithelial morphology. So as a little sneak peek of how these cells are going to look, we have the membrane stained with WGA and the nuclei stained with DAPI. You can see at least 2 U2S cells in this image here. So then we wanted to image our bacteria and our U2S cells together. So we took our E. faecalis and we grew it overnight. Then we took some of the culture and we added it to a well plate that contained the U2S cells. We let that well plate incubate for 30 minutes. We then removed the cover slips that contain those U2S cells out of the well plate and we fixed it for about 10 minutes. And again, this is following kind of the same procedure of fixation and staining. The only difference is we did add that WGA and let it stain for an hour before we washed and then mounted it and imaged on the confocal. So this first image here, we want to know how long until E. faecalis causes U2S cells to shed. So the shedding occurs when the bacterial cells invade the U2S cells. And when it invades the cells, it causes this infection, causes the cells start to kind of fall apart. And if it's in a monolayer, they start shedding off of that monolayer. So that's why it's called shedding. In these 3 images, we have U2S cells, U2S cells and E. faecalis. And same thing here, U2S cells and E. faecalis. This is after an hour of incubation and 2 hours of incubation. So we just have normal U2S cells over here. In this image, we can see some blue all along the edge of the U2S cells being the bacteria. So we can see that attachment after an hour. But after 2 hours, we actually saw some of the U2S cells start falling apart here on the edge. So we determined that 2 hours was too long for the bacteria and the U2S cells to interact, and that 1 hour was the better time limit. However, since we knew it took only 30 minutes for the phage to insert their DNA into the bacterial cells, we moved forward with a 30-minute interaction time. So that leads us into our next question. Can phage insert their DNA into E. faecalis strains that they don't lyse that are on U2S cells? So first we had to prep our bacteria and our phage. Then we combined the phage and the E. faecalis into those U2S cells and did the same prep as we did with the bacteria and the U2S cells. Then we imaged just the U2S cells on their own, U2S cells and E. faecalis, and then U2S cells, E. faecalis, and phage. So as you can see in this image, right here along this edge, and then right here is all of our bacteria. You can see the outline of the U2S cells. Same can be seen over here. We have bacteria all along this edge here, along the U2S cell membrane. And so with this imaging, we did see that bacteria near the U2S cells, but we did not see phage DNA insertion. And that led us to another issue here. So we had our green laser, and we could set it at either what we decided to set it at either 1% or 5%. And as you can see, we have this like increased green fluorescence for the 5% compared to the 1%. But regardless of either of these images, we still see this green fluorescence all across the U2S cells, which is not what we want to see and not what we expect to see. So we have this green laser problem. So as I said, there seems to be some green fluorescence across the U2S cells. So we were trying to figure out what was causing this issue. Is it the 1% or 5% green laser, autofluorescence, background noise, or phage? So we actually ran several tests to figure out if one of these was the problem. We still saw green fluorescence after testing all of these issues. So that led us to our final problem, which can it be unattached Cyto13? To test this, we took E. coli and E. faecalis, and we incubated it overnight for 24 hours separately from each other in their own cultures. We then took our E. coli and prepped it how we prepped it with the phage, how the phage were prepped. We combined it with Cyto13, mixed it end over end for 15 minutes, and then put it through a filter and measured the fluorescence intensity on the plate reader. We knew that the E. coli was not going to make it through the filter. So what should be remaining is just any unattached Cyto13 in that mixture. And when we measured the RFUs on the plate reader, it was below 1,000. So we took that E. coli cell-free solution, so there's no E. coli in there, and then the E. faecalis, and we added it to a cover slip, and then we went through that same process of fixing, washing, and mounting with DAPI. So what we saw with the 1% is we saw that E. faecalis had lit up here, the nuclei of the E. faecalis, and then we also kind of saw some green over here. However, it's much lower than, I promise you, it's much lower than what we have seen before. So we can confirm that it is not the unattached Cyto13 that is causing that green fluorescence. I promise it looks darker than this on other screens. And then we checked with the 5%, and there was this clear green fluorescence that matched up with the bacterial cells. So we figured out that it wasn't the Cyto13 that was causing this green fluorescence, but rather the laser itself. So first off, we know that the 5% green laser is not reliable for imaging, and that the green laser in general is picking up the autofluorescence across the U2S cells. But regardless of this, we know that there is bacterial present on the edges of the U2S cells. We were able to visualize that, and that there is no phage that have been seen. So that then leads us to our next section. Can E-facalis strains of interest attach to H-block cells? So H-block cells, and can phage insert their DNA into E-facalis strains so that they don't lyse that are on H-block cells. So H-block cells are human bladder tissue. They are spontaneously immortalized from normal urothelial tissues, meaning that they are not tumor cells. So they are spindle-like in morphology, and they are slow growing. When they are layered on top of each other, they can also differentiate. So I won't go through the whole protocol here, but we prepped our bacteria and our phage. I will draw your attention to this TMA-DPH here. So instead of using DAPI, we use TMA-DPH, which is a blue stain that stains the membrane of what it is, whatever it's interacting with. So in this case, the H-block cells and the E-facalis. And then we combined all of our mixtures into our plates that again had H-block cells instead of the U2S cells, and then we image on the confocal. So first we have H-block cells just on their own here. We have H-block cells in E-facalis, H-block cells in phage, and then E-facalis, phage, and H-block cells. So one thing you'll notice here is you have this ton of this green fluorescence. Again, that's due to the green laser over autofluorescing everything. So that's not actually true green fluorescence there. And then we were also able to make out all of these H-block cells, but we seem to have kind of an issue over here, as well as some issues with this blue fluorescence in the bottom left image. And that may have been due to a blue stain being added that we weren't aware was added, causing this blue fluorescence everywhere. So as I can see here, we have the nuclei and cell membrane are blue because DAPI was added, and I didn't know DAPI was going to be added. So we have this nuclei that is also stained blue because of that DAPI. Also, we did not take into our account that the length of interaction between the H-block cells and the bacteria. So since we didn't take into account, there was ended up being shedding of human cells. As you can see very clearly down in this image over here, a lot of debris of those H-block cells. Also, there was no consideration of the concentration of bacteria, H-block, or phage that was applied. So we weren't able to control that issue or control how they interacted. So we weren't positive how the phage was going to interact with bacteria and the H-block cells and vice versa between everything. But overall, we also did not see any phage staining. So I've given you guys, how this all works in more of a logical way, but that's not really how this protocol went. As Dr. Patanti kind of prefaced, this wasn't a great start in this project. So we originally started with this paper, Fast and Easy Phage Tagging and Live Dead Analysis for the Rapid Monitoring of Bacteriophage Infection. And this paper showed us how to image bacteria and phage together, both with the TEM and the confocal. They showed some great images between both of them. So we said, oh, cool, we can move forward with that paper. TEM did turn out great, but then the confocal, a little bit of a different story. So our first issue was that TMA-DPH, and as you saw, I only explained it once. It was only in that last set of images. That's because when I would look through the confocal to see how the stain was looking, it would bleach immediately. Like I could watch it bleach while I was looking at the cells. And because of that bleaching, we couldn't see anything when we went and tried to take images on the confocal. So TMA-DPH got thrown out. And because of how great the images looked on the in the paper, we also were able to confirm that we couldn't really replicate what they did because of the stain, and it was not reliable for use in staining bacteria and phage together. Another issue that we had is we started with the H block cells. So as you saw, that was our final image. We only were able to run one test with these cells until we lost access to them. And so we are scrambling to try to find cells to work with. Thankfully, Dr. Kanzok saved us with U2S cells, so very much appreciate it. We were able to have all the images that you saw today of the bacteriophage and U2S cells together. But while working with these cells, we ran into that other issue with use of Cyto13. And again, the issue isn't the Cyto13, it's the issue of using that green laser. So if we were to do this in the future, we wouldn't use a stain that emits light at a green wavelength. It would be either blue or red or another laser that might be available for use. So lastly, even with all of these steps that we had to take to scramble and finish everything, we were able to develop a protocol for visualizing phage attachment and DNA insertion into bacteria under laboratory conditions. We also were able to develop a protocol for visualizing bacteria attachment to U2S cells. Moving forward in the future, we would obviously use a different stain, one that didn't use the green laser, so we don't run into all the issues we did during the research. We would do more tests with different E. faecalis strains and phage to further confirm our protocols. And from there, we would expand to different bacteria species and their phage, as well as different cell lines. I have several people that I'd like to thank. First off, Dr. P. I very much appreciate. Oh no,
I won't look at you.
Or get some water. I very much appreciate all the support and patience that you provided during this entire research. Without your help, we wouldn't have been able to go from plan A to plan Z and still give you guys research today. So I very much appreciate it. Dr. Mearsh and Dr. Dale. Without Dr. Mearsh, I would have not known how to use a confocal at all. Oh gosh. And so all for patience and time to figure out how that works was very much appreciated. And of course, Dr. Dale, for all of our conversations that we had to keep me on track and not start freaking out and trying to figure out what I should do next. I very much appreciate that. I thank you to Loyola University's biology program for the funding for this research. I have several professors here that I'd like to thank. I'd like to thank Dr. Kanzok for saving me with the U2S cells, as well as the time to prep those cells when there was no one available to make them. So I really appreciate that. I would like to thank Dr. Pickett and Paula for feeding me when I refused to eat, as many of you know, as well as our great conversations just on life in general and helping me with this research. I'd like to thank Dr. Grande for helping me stay on track and get my research done, even with the setbacks we had. Dr. Kula, Alex, I can't say Dr. Kula, for your help whenever we were having issues with any sort of contamination. You helped me figure that out. Dr. Wolf for providing the bacterial isolates that I worked with, and Aditi for the time that we did have testing with the H block cells. I very much appreciate her help with that protocol. And then of course Joe for his help begrudgingly on the TEM. Without his help, we wouldn't have seen those beautiful images that I took from the TEM. I'd like to thank the Tonti Lab and other MS biology students. Without your guys' help, I wouldn't really had that camaraderie that I needed to get through this research. And the distractions that were needed, you know, when I needed to take a break. I'd like to thank my family for all the love and support. During this entire time here, without their help, I wouldn't have had the confidence to complete this research. I'd like to thank a few friends. I'd like to thank Liz. She is actually in a math biology master's program, or math MS program. So she had the unique experience of being in the same kind of places I am. So we were able to complain together and, figure out our own issues as we work through our research. She's finishing up hopefully next year. I'd like to thank Claudia for being a great roommate and dealing with all the rants that I would go on, especially recently. And then I'd like to thank Emma for allowing me to crash the lab, steal her time, use her entire bench, like just to prep all of my confocal work that I had to do. So thank you, everybody.
Question.
Ellen.
Okay, 2 things. One, congratulations on your Lions win last night. Very, very happy to hear that. This is in relation to your beautiful SEM, or no, not SEM, fluorescence images. They were gorgeous, and I had a very biochromatic question about them.
Oh.
Is there a way that you could look at the different amounts of fluorescence, especially when it comes to that green laser, that phage DNA, like pixel by pixel, you know what I'm saying, and computationally confirm it?
Well, I mean, I know we probably could. I don't currently have that knowledge on how to do that. However, on the microscope, you do have the opportunity to change like the percentages as well as like how much additional gain there was. So we can adjust how much is on there. And we felt like the settings we have here were best in order to get the images, that we have without oversaturating with the green stain, even though that still happens.
Was there do you think there was still that green noise happening when you were just looking at the phage isolated?
I don't think so as much. We were pretty confident that that was actually the phage DNA insertion.
Yeah.
And that's because we didn't have like it where it was like covering the entire cell and it was very concentrated within those bacterial cells. So we felt that that was clearly like the phage DNA getting inserted into the bacteria.
Yeah, that makes sense. Thank you.
Yep, Dr. Kinzog.
Very nice. For your first screen that you showed us for like the plus and minus with the red and the green boxes. So me not being a phage specialist at all.
Yeah.
When they show those plaques,
Yep.
I think are they all like the same size, or are some of them like slower and some of them faster? And if you see those plaques, I'm just curious in terms of kind of those host phage relationships. When you look at the yellow ones, I think are
the ones that have
the
plaques.
plaques. Are they like some smaller?
Yeah, they
larger,
can vary. We had our myovirus one actually was usually a bit smaller. We had trouble with it getting it to a high enough concentration to get bigger plaques, so that does change how big the plaques are. Usually it still takes overnight for them to plaque regardless. But yeah, that's usually about the variation we can see. We can also make them smaller if we'd like to do a dilution, depending on how much phage we add. In the one that you saw here, I added about 10 microliters into one spot versus slowly diluting, so there's less and less phage. Because that was several phage together rather than just like tiny individual plaques. There was just too many to fill out a hole. Yeah.
Lovely presentation, of course, but this is kind of a silly question, but you mentioned like earlier on that you mounted it with nail polish. Like how did you get to there?
Oh, it was definitely for the smell. No,
I mean,
no, the nail polish helped make sure that it could remain like contained somewhere for a long periods of time. Because there's this issue that maybe the cover slip will slide or will like lose bleaching or staining, stuff will bleach. And so using nail polish to seal it, make sure we don't have any of those issues when we go to image. Yeah, Ellen.
Sorry, I have another question. When you brought up this host range, which is so interesting that you saw that insertion in those ones where you didn't see lysis. Do you think that they're just like depending on the host, they're in the like lysogenic cycle where.
I don't think it's that, because I think there can still be like, as we saw, we had like no attachment from myvirus one, but we saw that phage DNA insertion. I think it's because that the oh my gosh, I lost my train of thought. Since the phage is getting inserted, that doesn't necessarily mean that it will then kill the bacteria, because there are defense mechanisms that the bacteria can use to maybe get rid of it once it's inside of the bacteria. So just because we saw that insertion, that doesn't mean there will then be killing of the bacteria cells.
Yeah.
Yeah, like great presentation. Do you guys do any follow-ups with sequencing the bacteria? Because wondering if failure to see an attachment of a phage to what it's supposed to be exposed could be evidence of the bacteria actually undergoing some mutations, like
Yeah.
spontaneous mutation.
Gosh, I wish we did. With those issues with all of the staining and stuff, we really were just kind of getting towards the end when we actually did all those host range tests. So unfortunately, we weren't able to. It would be great to. That would further explain kind of what we were seeing in our images. But yeah, no, it would be a great thing to do.
We do an online question.
Yeah.
Emma, do you want to turn on your microphone?
Yeah, sure. Okay, so I said that it might be a dumb question and I don't even know if it's related, but when you talked the slide that you talked about something causing the cells shedding, you might have said it, but I was just curious, like if you knew how that kind of happened. That was pretty cool. Like, was it from the thing that you inserted into the cells, or like
So it is.
past it?
Yeah, it's this one. So usually the bacteria are able to invade intercellularly into the U2S cells or whatever cells it's working, human cells it's working with or interacting with. And it can cause, like with patients, when you get this infection, it causes those cells to kind of start falling apart. And so that process, again, I don't know fully the chemical, reaction that or whatever happens in there, but overall, that infection causes those cells to kind of start falling apart. You could kind of see it in the far right image where their cells are kind of starting to like stretch apart because they're starting to separate from each other. So just that bacteria getting into the cells causes that.
Got it. Thank you.
Mhm.
Good job.
Questions? No? All right, well then let's thank Ally again. Parting words to you are, eat this food. I do not want to carry it back across campus. You all have pockets. Getting lots of
Notes.
Okay. Maybe come to you. That would be amazing.
Oh, it's already.
I was actually.
Yeah.
Good job, Allie.
Thank you.
Yeah.
So.
There.